maybe wait a couple of minutes. And I'm gonna be recording the webinar as well. So we're gonna wait a few minutes for attendees to join our meeting. Okay, it's 2:33. I think we should start now officially. So let me welcome everyone. This is the third session of the Applied Ecology Minor Research Symposium. So thanks everybody for joining and for supporting our students and our program. This afternoon, we have 3 students that are going to be presenting. Each student is going to be presenting for 10 to 12 minutes. After the presentation, then we will have time for questions, maybe 3 to 5 minutes at the most. So please take notes on your potential questions so you can type them in the chat at the end. If you if you have a question, just type it in the chat. We could also unmute, or I could also unmute, but the chat seems to be like the most efficient way. And just notice that we're recording the session. So with that, let's get going. The first presentation then. Let me get my list. The first presentation is going to be by Sarah Rakita, and the title of her presentation is Water Quality and Environmental Justice. So Sarah, please share your screen and your presentation.
Sorry. Mm-hmm.
Yeah, don't worry, take your time. We're waiting for more attendees to join, so we're good.
Okay, should be.
I can see it, so you're set.
Perfect. Thank you all for coming today. I'm super excited to share with you all what I've been working on this semester and to hear from the other students as well. So to begin, I'll just briefly introduce myself. My name is Sarah Rakita. I'm a junior here at NC State. I'm majoring in environmental science, and I have 2 minors in applied ecology and environmental toxicology. I'm interested in hydrology, toxicology, human health. So this project really aligns with those things. And it's been super interesting to kind of learn more about those topics and how I could apply those to kind of future career goals and those kind of things. So today I'm going to be talking to you about water quality and environmental justice. Throughout the semester, I've worked with an organization called Crowd the Tap. Crowd the Tap is a citizen science project that's funded by the EPA as well as NC State. And so their main goal is to kind of educate individuals about the type of water pipe material that they have, as well as some outreach programs to just get people more educated on their drinking water quality. And then they strive to kind of talk about infrastructure replacement of those pipe materials. And so our main focus currently is kind of through high school students. So we partner with high school teachers, and then they kind of educate their students about different water quality aspects. And then the students have the opportunity to go home, look at their own home pipe systems, as well as utilize water testing kits and then get information about kind of like their water quality. And this is throughout the entire United States. So it's been cool to kind of see that throughout the US. So before we get started, I'll just talk a little bit briefly about water quality and environmental justice in general. So some of the big drinking water regulations that we've kind of been focusing on recently is the Safe Drinking Water Act of 1974. Currently, the Safe Drinking Water Act is overruling of 90 different types of contaminants for drinking water. So it just kind of regulates 90 different contaminants to make sure that drinking water that you have is safe and isn't infiltrated with those contaminants. Of course, there are violations that happen to this, which I'll talk about in just a second. And then the next one is the lead and copper rule of 1991. This was really important in establishing something called the maximum contamination level goal, or the MCLG. And so the MCLG just kind of just identifies different contaminants and the maximum amount that would be deemed kind of unacceptable within your drinking water. And so in specific, it talks about lead, and we have a zero tolerance rule for lead. There's no deemed amount of lead concentration in your drinking water that's acceptable to human health. And so obviously we still are finding lead in drinking water. There's still a lot of lead pipe infrastructure. So this rule is in act, but it's not really upheld very well. And then environmental justice in general is just the unfair exposure of environmental burdens on poor and marginalized groups. So it's looking at how, our environmental degradation is really affecting those groups of marginalized communities. So the objectives of my research this semester was kind of, where is our current water testing coming from? So what parts of the United States, of North Carolina, have we really outreach to through Crowd the Tap? And are we including these marginalized groups within that testing? And then are we seeing more lead exposure results in these marginalized groups? So is it apparent that lead exposure is happening more often in these communities of lower economic statuses and marginalized groups? Okay, so because this is a citizen science project, a lot of the data collection is actually done by the general participants, the students, their families. But kind of the main thing that we get from that, we do a lot of survey collection, so they answer some general questions. We currently have around 2,000 data from people, different individuals. Around 2,000 of them have answered kind of these surveys and some water samples. And so kind of my main focus this semester was just sorting through a lot of that data, doing a bit more data analysis than anything else. So on the survey, we kind of asked just general questions of their home type, whether it's like a standalone home, rented, other aspects of their insurance, their race and ethnicity, as well as the kind of service and home pipe material that they probably have for their drinking water. And so obviously with surveys like this, there's definitely going to be bias, non-response biases, those kind of things. So you have to be careful with that going forward. We also get the water sample results. So kind of the second step in the program is taking actual water testing from your home tap. And so we give them kind of the basic information on the correct ways of doing that, like flushing the water out for about 2 minutes and then taking those. And then those are sent to a lab, I believe in Virginia, and then we get the results from those. So our main focus is definitely lead, but it also screens for about 28 other kind of elements, some metals, some organics, and those kind of things. And then the main thing that I was kind of doing methods-wise this semester was using ArcMap and GIS applications to make some maps of general participation, as well as kind of like the overlap of the water quality that we're seeing and environmental justice aspects. Skip the slide, there we go. Okay, so the results from this. So this map, I'll start by apologizing quickly. The maps are a little easier to see on a full screen, but of course for this presentation, they needed to fit in the slideshow. So I apologize for it being a little grainy maybe. But this is just our general participation throughout the United States. And so we kind of didn't know where we were even getting a lot of our data from. So it's just a general gradient of the colors there. The darkest reds are where we have a lot of participants, and the lighter pinks, reds are where, we don't really have much information. So actually 45 of the 50 states are representative. So we only have 5 states where we don't have any participants. So I would say that's kind of a good starting place. You can see, North Carolina, Ohio, California have some pretty dark red. So that's kind of where our biggest outreach currently is. And then we dove in a little bit deeper to North Carolina specifically. So on a county level is what this map is showing. So again, this was just giving ideas of general participation on this county level. And you can see kind of in the middle there, the darkest blue, that is Wake County. So that's where we currently are. NC State being, a big funder for this program. We have a lot of outreach. We've connected a lot with the local school systems. So it kind of makes sense that we're getting a lot of data from these places. But you can kind of see throughout, we have data from all the different counties. So that's definitely good as well. So then with this information, we kind of wanted to look at, where are these marginalized groups and are they even representative in the data that we have? Can we even determine anything from the data that we have? So the next map that I kind of put together, again, this one's a little bit harder to see with how small it is. But I compiled this map. So the back map of North Carolina is actually split up into our US Census block level here in North Carolina on a standard of poverty and then race and ethnicity. So basically it's showcasing underserved communities. And so the standards are kind of listed there and you can read over those. But anywhere that it's filled in with the purple is saying that it is deemed an underserved community based on these standards here. And so then I went through our current data in North Carolina, and the red dots are where lead was detected in households that I that identified as people of color. And so you can see, again, that big cluster in the middle is from Wake County. So of course, a lot of our data came from Wake County. So we definitely have some there. But then it's kind of a little bit more sparse throughout. And then you can continue to see that a lot of these red dots aren't really on top of any of the purple locations. So that kind of showcases the idea that these underserved communities aren't really getting this outreach program from Crowd the Tap. And so then you have to kind of think about, is our data really representative of all of these communities? So just some discussion about that. As I was just mentioning, I think the biggest thing is to continue the testing and the outreach and the education to these marginalized groups. And as we saw, we're kind of missing out on those a little bit. And I think it would be really important to incorporate those communities, especially from past research and articles about environmental justice. We do see that there's kind of a connection between these groups of people getting affected more by these environmental burdens. So it would be really interesting to see if that lead contamination is more prevalent in those areas. And then kind of the next thing going forward would be a little bit more statistical analysis, some hypothesis testing. I was kind of hoping to do a little bit more with this, and hopefully maybe in the future I can kind of continue to do that. But looking maybe at things like housing security, socioeconomic factors, insurance coverages in these communities, and kind of seeing how these do or kind of don't connect with one another in those areas. And then just an acknowledgement quickly, I just want to thank Dr. Karen Cooper. She's the principal investigator of this program here at NC State, and she's been super helpful, just giving me more information about water quality and environmental justice in general, and then obviously helping me with the research. And then special thanks to both Nicole and Emma. Nicole was working with me at the beginning of semester as the project manager for Crowd the Tap, and then she got a new job. So then Emma kind of filed in and helped me towards the end. So special thanks to both of them for kind of helping lead me through this whole process. Yeah, that's the end of my presentation. Thank you all so much for listening today. I really enjoyed sharing with you, and if there's any questions, I'd be more than happy to answer them. Thank you.
Excellent. Thank you, Sarah. Great presentation. Very interesting. Again, everybody, you guys can type your questions in the chat, and Sarah can read them, or I can read them for her. So feel free to enter any questions. So I know that you did not participate much on the citizen science part of it, but I would imagine that's probably complicated to get people to participate evenly over the state to really come out with a very quantitative assessment for North Carolina. Any ideas on what type of limitations the team had in reaching people and getting them to join?
Yeah, definitely. So I know personally, I was kind of asked to reach out to, my old high school teachers. I'm from Ohio, so, I was reaching out to people in Ohio. So it's kind of just like word of mouth at this point, which is kind of difficult to, reach areas that maybe you're not familiar with. I know there was like a money incentive for teachers that were participating. So they did get some monetary money there. And so, that was probably helpful for some people to participate. But I definitely think it's just kind of hard to outreach to them. I think what's good though is that we do provide a lot of educational information. And I was fortunate enough to kind of send out the results back to people. And so that was really interesting as well to talk to families a little bit more kind of about their results and what we were finding.
Right. Yeah, that's great. Okay, let me just make sure that I have the settings right for the chat. I think so. I think people should be able to chat with everybody. Okay, any questions from the group? Okay, apparently not. So, I mean, it'll always take some time for people to respond, but I think we can safely assume that there are no questions. So thank you, Sarah. And so let's move on to the next presentation. Now we are going to be listening to Hannah Werner, and she's going to be talking about stable isotopes in Hawksbill sea turtles.
Hi, my name is Hannah Werner. Just to give a brief overview about who I am, I'm a junior at NC State University. Right now I'm majoring in animal science with a concentration in pre-veterinary bioscience. I have my minor in applied ecology, so that's where this research focuses in on. I did my research this semester on stable isotopes in Hawksbill sea turtles. So looking at the Hawksbill sea turtle, we know that they are a critically endangered species. What we do know about their diet, we don't know too much, but we do know that they're omnivorous. They eat a lot of sponges and a lot of seagrasses. They are also known to be capital breeders. So what that means is that when they're at their foraging grounds where they are eating on those sponges, seagrasses, whatever it may be, they are building up an energy capital. And that energy capital is then used for when they migrate to their nesting beaches, where they then reproduce and they lay their eggs. So that capital breeding area is very specific to them in the way that they're building up that energy, then using it, and that's how they use it to reproduce. So looking at our population of interest, we focused on Antigua. So as you can see with this map in that upper right-hand corner, you can see where Antigua is. It's in the Caribbean Sea. And just to give you a closer view on Antigua, our population of interest was at the Jumbie Bay. That's an island off of Antigua. It's important because it has a very large population of nesting sea turtles, specifically the Hawksbill sea turtle. So that's why we focused on that area. So looking at our population of interest, again, we focused on Antigua, that Jumbie Bay region. We had 90 individuals. They were all nesting turtles that we sampled from. We took multiple samples from these individuals, then sent them out for analysis. Our population decline, that's what we're noticing based off this map. So this map right here, that was formed from the Jumbie Bay Hawksbill project. They've been monitoring the nesting turtle population since the year of 1987. As you can see, looking at this graph, in the most recent years, we have a decline in population, more so of the nesting turtles. So this could be in relation to two different things. We could be noticing this kind of trend based off of less turtles, like I mentioned. they are critically endangered. So maybe we're just noticing less turtles because they are dying off just because of environmental reasons, since they are critically endangered. The second reason could be they're reproducing less. So reproducing less could be related to that capital breeding thing I talked about earlier, how we have that energy capital they build up in their foraging grounds, and then they go off and they reproduce on the nesting beaches. So if they're eating less or eating a different diet, are they not getting enough capital to build up to be able to move on to these beaches and reproduce more? So that's kind of where we move into our stable isotope conversation. That's what we focus on this semester for. So the stable isotopes, you are what you eat. What that means is that when they're consuming this specific diet, it goes and it's incorporated into the consumer cells. So it's incorporated into skin cells, bone cells, and blood cells. We focused on skin cells of the Hawksbill sea turtles. So we're able to look at those ratios within the skin cells. The ratios are based off of discrimination that naturally happens within nature. So we usually see a discrimination against the heavier isotope. So on the right side here, we have the carbon 12 versus the carbon 13. That is one of the ratios we looked into. The difference between the two is one is heavier. That would be the carbon 13. It has one extra neutron compared to the carbon 12. That's what makes it heavier in mass. So when we see those differences in nature, we're able to form a ratio that is specific to the diet that the turtle is eating. Then we're able to look at these ratios and make inferences about the diet and base those off those unique ratios. So like I said, we looked at carbon 12 versus carbon 13, that ratio in specific, and then also nitrogen 15 versus nitrogen 14, that ratio is also very important. And then, like I said, it relates to foraging ecology. So you are what you eat. We can look at that and discover the specific diet and make inferences about the diet. So moving on to our null hypothesis, based off of previous data, what we would expect to see is consistent stable isotope ratios in the carbon and nitrogen. That would indicate that the individuals within our sample population of the Hawksbill sea turtles, those 90 individuals, we would expect to see that they're migrating to the same place and consuming the same diet over time. So then looking at our field methods, a group of people went out to Antigua. They stayed off the nesting beaches on that Jumbie Bay region, and they were able to wait for the turtles to come up to the nesting region. Once those turtles got there, they used that opportunity to take a skin sample. And once they had that skin sample, that's what we analyzed in the lab. They also had these satellite tracking devices implemented into the turtles. We don't use those, but those are very important for the future implications of this research. Then for the lab methods, this is where I come in. I took these samples, they're housed in these little containers. The containers are important because they help with contamination. They keep contamination out, as well as the use of gloves and sterilizing all the equipment before putting the samples onto the equipment. So that's what these little containers are super helpful for. Then once we got the sample out of the container, we put them into these little trays. These trays were helpful in allowing us to soak the sample in distilled water. So distilled water was important because not only does it get rid of the salt was used during storage, so it rids us of the salt, and it also helps us soften the sample up. So once we have the sample softened, I was able to use that little mirror and razor blade set up over there to cut up the sample into fine particles. Once I had the sample into fine particles, I was able to put it back into the tray. So we have the sample back in the tray now, and then we were able to put this tray back into the oven. Once we had it in the oven, we were able to dry it up a little bit, and drying it up allowed us to weigh it more. So using this setup here, I was able to take out those drier, finer particles and weigh them on this scale here. So there are tin capsules in this photo. Using the tin capsules, you're able to weigh the tin capsule, get that weight, tear it out, and then weigh the fine particle sample. There had to be a specific weight requirement for the sample before sending it out to the lab. So weighing it allowed us to make sure it was within that specific requirement, and then we could send it off. So once we had everything weighed out, we would use this little metal tray there with all the instruments. We would use those little instruments to fold up the tin capsule onto the sample. We would make sure it was nice and compact. We didn't want any holes, again, because of contamination, and we didn't want any of the sample to fall out, just because we needed that weight requirement to be consistent. So once we had our samples all packed in and ready to go, we were able to put them into this pink little container there. That pink container was then used to send off to the lab. And they went to the Yale Mass Spectroscopy lab. The mass spectroscopy lab is super important with stable isotopes. What it does is it basically combusts our samples, and it changes our samples from that solid particle into a gas particle. Then using that gas particle, we're able to look at the differences between the weights of the two. So I mentioned the discrimination between the heavier and the lighter isotope. We're able to see the heavy isotope and then the lighter isotope, and then look at the ratio between the two. And again, we mainly focus on the carbon and nitrogen elements. So that's how we got our analysis back. And once we got the data, we were able to look at what the data looked like. So again, our sample size, we had 90 individuals. We focus on these individuals in specific because we were able to take samples multiple times from these individuals over the years. So we had about 245 samples sent to the lab. We then use program R to make a linear mixed model. This linear mixed model is super important for our data because it allows for that individual variation is what we're focusing on. So what it does is it allows for each individual turtle to have their own y-intercept, but then also make all these individuals into one slope. So then that way we don't have some weird crazy graph like this. So we have this as our delta nitrogen, which is the ratio of the delta nitrogen 15 and nitrogen 14. So looking at this graph right here, this is our raw data. So each of these lines is an individual turtle. And as you can see, looking at this, each individual turtle has its own slope and has its own y-intercept. Looking at it, you can't really see a specific trend. You can't really make an inference from it. You can just see there's a bunch of chaotic lines. So we were able to use that mixed model to use this raw data to produce a model fit, which is what our final data looked like. So this is our nitrogen ratio here. As you can see, the most important takeaway from this slide is we're seeing a positive increase. We do have a lower p-value, it's lower than 0.05, so that way we can see that this is statistically significant and we're allowed to accept this data to go against our null hypothesis. So again, we had this positive trend over the years when looking at our nitrogen isotope. Then going to the carbon, again, this is how we fit our linear mixed model. We had this raw data set where we had these crazy lines going throughout. Each individual line is an individual turtle with their own y-intercept, their own slope. So looking at this, you can't see a specific trend. You're not able to make an inference from it. So we're able to refine that and produce a model fit. And that gives us this perfect carbon graph that shows a decline over time. So our p-value here, it was actually lower than our nitrogen. So this one's actually a stronger relationship here. And so again, since it's lower than 0.05, we're able to accept this data as well as being statistically significant. So what does this data mean for us? How are we able to interpret this data? As you can see from the changes on those graphs, we have that positive change in nitrogen as well as the negative change in carbon ratios. So the carbon is important because it could potentially be related to the Seuss effect. The Seuss effect doesn't naturally explain the effect of the carbon, but it could be a potential explanation. So what the Seuss effect is, it is slow changes in the carbon makeup of marine organisms over time, and it's made from the carbon dioxide emissions into the air that then go into the water. So looking at that, with this Seuss effect, we usually see a decline in carbon over time in marine organisms. So looking at our graph is very consistent with that data. It's not the specific explanation, but it could be a potential explanation. And then looking at our nitrogen ratios, it again goes up. So that's a change over time. Our hypothesis said we shouldn't have seen a change, but we did. So that relates to our foraging grounds as well. So coral reef health, we know coral reef health has been a big thing in the news for the past few years. It's been changing rapidly. So since our turtles usually stay in those coral reef areas, if those coral reef areas are changing and their environment is changing, then overall we can assume that their foraging habitats are changing as well. And so with that, they could be eating something different. They could be going to different grounds, or their environment is just completely changing on top of them. So all this kind of funnels down into the population decline. And the main reason for that is that big change in foraging grounds. And that's why we're seeing those changes in our carbon and nitrogen ratios. So then with our discussion for our future implications, at the top of this page, you can see that there are some pictures for satellite tracking maps. So with satellite tracking, I mentioned that very briefly earlier. We can use satellite tracking to see specifically where the turtles are going and what they're eating. We don't know specifically where they're migrating to eat and then where they're going back. We just know how to identify their nesting beaches. So by using satellite tracking and using those nesting beaches, we're able to see where the turtle is going from the nesting beach to the foraging grounds. And by identifying those foraging grounds, we're able to identify and monitor their foraging habitats. And so that way we can look into specifically what they're eating and how they're living in that specific environment and decide whether or not that environment is completely changing or what else we're noticing within the environment. That would help us discover more things about the population and make more inferences about their diet and why the population is decreasing. And at this time, I would like to acknowledge Dr. Moyer for helping mentor me throughout this whole project. And then of course, Dr. McKinney for giving me the opportunity to work with Dr. Moyer. And at this time, I'll take any further questions.
Thank you, Hannah. That was a great presentation. Really interesting topic too.
Thank you.
Let's see if we get any questions. One that I was wondering, the nitrogen increase and the carbon decrease, but it was a small change. It wasn't really large, right?
Mm-hmm.
Are there other studies that you can compare with to see whether the magnitude of change that you saw was kind of like unusual or within kind of like expected changes?
So with the null hypothesis, what we basically based it off was previous studies. So Dr. Moyer, who I worked closely with, did a lot of research on this for the past few years. And these have been, the stable isotopes ratios have been monitored by multiple people. So looking at the decrease over time, our main focus is on the individuals that we're looking at and at our population. So again, we need to do further research to examine why those changes are happening. We don't know too much about their foraging habitats or what they're specifically eating. We know they specifically eat sponges, but we're not too familiar with the chemical makeup of those sponges, which is what makes this difficult. So one other further thing we could do is, since the ratios are specific to the source that they're eating, is comparing it to specific sources that could be a potential diet to those turtles. But since we're not too familiar with that, there aren't too many questions that could be answered based off of this data just yet, which is why we need to do more research.
Right, yeah, that's very interesting. So let me take a look at the chat. I don't see any other comments, so we can probably stop here and move to the next presentation. Thank you again, Hannah.
Thank you.
You got a comment there that says, great job. Okay, so the next presentation is by Claire Melvin, and she's going to be talking about Echinacea labiegata, resource allocation and trade-offs.
Hello, my name is Claire Melvin. I am a senior in natural resources with a concentration in ecosystem assessment, and I worked in collaboration with the Irwin Lab and the Applied Ecology Department to focus research on a federally endangered plant, Echinacea labiegata, also known as purple coneflower, and I will be referring to it as Echinacea for the rest of the presentation. And I focused on research, trying to understand how resources were allocated across plant production in both vegetation growth and reproductive output to help understand the reproductive biology. So it's important to understand the habitat that Echinacea is found in, and they are found in prairie communities that were once very common across the Southeast United States and were historically maintained by disturbance of fire and grazing. And due to human impacts like settlement and lack of disturbance, these plant communities are in decline. So today, you can find them in areas that are disturbed and maintained by chemical or manual disturbance such as mowing and herbicide use in roadsides and right-of-ways. And this includes a power line right-of-way pictured here. This is Picture Creek Diabase Barrens, and it's in Granville County, about 45 minutes north of NC State. And you can see the high levels of sun and the open canopy that really allow for high levels of plant and herb diversity and high levels of diversity in pollinators. And in Picture Creek specifically, there are about 50 rare plants associated or occurring here. And this includes the largest population of Echinacea labigata, which is a federally protected plant that's being considered for downlisting as of last year. And this plant, although it's meeting its recovery goals and populations are being protected and maintained, there is a lot that is left to be known about the long-term trajectory and how these populations are actually doing, including their reproductive biology. And so we know that these plants rely on seeds to reproduce, but in general, plants have limited resources due to their stationary nature, and these resources are required to be allocated across the plant. And so trade-offs can exist. For example, larger plants, they'll put more energy into growing bigger, and so they might have less of a reproductive output, so have less seeds produced and produce smaller seeds. So we know that Echinacea has the potential to produce many fertile seeds, as we can see in our picture over here. All of these light brown spots are seeds, but there is a difference between the actual seeds that are being produced and what's actually growing. And we don't know what's controlling the seed production in the first place and why multiple seeds are being produced or how they're being fertilized. So to understand the resource allocation in Echinacea labigata, I looked at trade-offs in reproduction by testing 3 hypotheses, including higher seed production will result in a smaller seed size, larger plants will produce less and or smaller seeds, and seed weight could indicate fertilization because large seeds are more likely to be fertilized. So first step in this process was collecting morphological measurements in the field at Picture Creek Dye Base Barrens in 2021, and the seed heads were collected along with different measurements. And these seed heads were then cleaned and the seeds counted to obtain a seed count per head. Once that was completed, 60 seeds were randomly selected from the seed heads, and these 60 seeds were individually weighed to obtain an average seed size per head. So we know that these plants produce a lot of seeds, but when they're producing a lot of seeds, will they be producing few large seeds or many small seeds? And I predicted that higher seed count will result in a smaller seed weight and seed size because they're investing more resources in producing more numbers of seeds versus larger seeds. And we can actually see that there is instead of a trade-off, there is a positive correlation between higher seed count and seed size. So when more seeds are produced, they're actually, Echinacea is actually making larger seeds as well. And this positive correlation, I wanted to see if it would correlate between vegetative growth and reproductive growth. So is there a trade-off between vegetative growth and reproduction, or is it another positive correlation? So I predicted larger plants will produce less seeds because more resources are allocated to vegetative growth. And I compared the morphological measurements taken in the field that are proxies for vegetative investment, including the height of the flowering stalk from the base of the plant to the top of the seed head, the number of basal leaves, and the length of the longest basal leaf. So instead of a trade-off, the most indicative morphological measurement was head height, and there was a positive relationship between head height and seed count. So these taller plants are actually producing more seeds as well, and therefore larger seeds too. So instead of a trade-off, these reproductive aspects of echinacea have actually been correlated positively. And these large seeds produced by large plants can be indicative of fertilization, because large seeds are more likely to have been successfully fertilized. Larger seeds have a higher chance for germination and fertilization. So because of this, fertilization can be a measure of maternal fitness. And I predicted that seed size and that larger seeds would indicate successful fertilization, and therefore have an insight into what the maternal fitness of the plant is. And I actually determined that seed weight alone is not enough to understand successful fertilization in echinacea, because this diagram shows 3 different seed heads with the 60 individual weights of each seed, ordered from lowest to highest across the x-axis. And so looking at the first seed head in the graph on the left, the red line indicates 0.0018, which was a good cutoff between fertilized and unfertilized seeds when looking at the data set as a whole. So it can be assumed that the seeds in seed head 308 were not successfully fertilized. And looking at the middle graph, seed head 512 has a very clear differentiation between 2 different groups of seeds. And it can be presumed that the seeds weighing less were unfertilized, and the seeds weighing more were successfully fertilized. But the graph on the right shows a continuous distribution. So without this clear differentiation between unfertilized and fertilized, looking at the weight, we cannot determine the percentage of successfully pollinated seeds in each head, because there isn't this clear delineation between unfertilized and fertilized seeds. So moving forward, it would be important to include germination trials of these seed heads to, again, assess maternal fitness and seed viability. So looking at these results together, larger plants are actually producing more seeds and larger seeds. So instead of seeing a trade-off between the vegetative growth and reproductive output, we actually saw a positive correlation between these 3 reproductive structures, including the seed weight, seed count, and height of the flowering stalk. So we will reject hypothesis 1, and higher seed production will actually result in larger seeds. Hypothesis 2, where taller plants will actually correlate with higher seed production, and reject hypothesis 3, where seed size is not enough to determine successful fertilization. And moving forward, this contributes to our understanding of seed production in echinacea. And we already knew it wasn't limited by pollen and soil cation concentrations. So now we understand it's not limited in the vegetative investment of the head height, of the flower. So what is actually limiting seed production and limiting fertilization? It could be natural selection, genetic differences between plants, or the microsite conditions that these populations are found on. And further research could look at trade-off between head height and the other vegetative growth measurements, the other morphological measurements. And it would be really important to evaluate these same plants across different years, and pollination studies will further help understand fertilization, and germination trials are needed. And this can all help inform our understanding of maternal reproductive investment. So then we can understand in the future how this can correlate with maternal fitness, which will help us learn how these plants are doing in the long run and their long-term projection and their, yes, thank you. I would like to say thank you to the Irwin Lab and Aaron so much for their help, and Trey, Dr. Becky, and Dr. McKinney, of course, played a huge role. And any questions?
Awesome. That was a great presentation. Thank you. So again, people can type their questions in the chat. There's already a comment there. So the question is, do you think that there could be any confounding variables that might affect the correlation between seed size, plant size, and the number of seeds?
Definitely. I think when you're working with plants, there's this long-term study, and perennial plants, especially, they vary between, they can vary seed production wise from year to year. And these long-term experiments can have a lot of human influence in them and in the experiments. So confounding factors could be data collection in the field alone, but then also looking at the environment that they're found in and the resources that are available to them. And is it, you know, we saw those sunny conditions. Is it the high sun areas that they like? And echinacea can also grow in forested areas. And that could be an example of a confounding factor too. So definitely, thanks.
Great. So I was wondering, there are other species of echinacea, right, that are like even commercially important. Did you get a chance to compare how, you know, the traits versus the one versus this species that you're focusing on?
Not necessarily, but yes, I think it is important to note that those species that are commercially available are found in a lot of places, and I wonder if they're supported just by the landscape industry. I don't think there's much difference between the pollination of the two of the different species, but it would be interesting to look at the differences between the commercially available species and this rare plant species. And I do know that Echinacea pallida is one of the more common ones that you think of when you think of coneflower, and it was actually state listed up until last year, and they delisted it. But yeah, it has made a lot more progress, I think, than Echinacea flavigata in terms of population growth.
Right. That's very interesting.
Definitely, thank you.
See, unless I don't think there's any more questions. So thank you, Claire.
Thank you.
So let me just thank everybody for those great presentations. It was a very interesting set of presentations today. And this ends our symposium. So we had 11 presentations total, 8 on Friday and the 3 that you just listened to today. So thanks everybody for supporting our minor students and their great research. So with that, I'm gonna stop recording.